Good morning. I am your host, Facundo Batista. Together with Richard Young and Lena Afeyan, we have organized this MIT course on COVID-19, SARS-CoV-2, and the pandemic. The basis of this course is to learn from leading scientists what is known about the new virus and the pandemics today. To this end, I have the great pleasure of introducing a wonderful colleague of mine, Professor Shiv Pillai. Shiv studied medicine and completed his PhD in India, and as a young scientist, he joined the laboratory of Levi Baltimore, one of our previous speakers at the Whitehead Institute as a postdoc. Over the course of his career, Shiv has made several important scientific contributions to immunology. As a result of this, Shiv is the Professor of Medicine, Health Science and Technology at Harvard Medical School. He's also a valued faculty at the Reagan Institute, where he runs as a director an NIH-funded Center for Excellence in Autoimmune Disease. However, what makes Shiv distinct is his real passion for teaching and mentoring young scientists, which he has done over the course of many years. He is indeed the director of the Harvard Immunology Master and PhD programs. He's also the director of the HST MD research programs. And over the course of his career, he has received several teaching awards that include the Irving London Award for Teaching, the Thomas McHone Mentoring Award, and he has been listed as one of the Harvard's Crimson Professors of the Year. Finally, Shiv has a secret witness from RAP, and if we are lucky enough today, we will be hear not only science, but one of his reasons at the end of his talk. So thank you very much, Shiv, for joining us. It's a pleasure to have you with us, and we are very much looking forward to your talk.
Thank you, Facundo, and thank you, Rick, Facundo, and Lena for putting together this course. This has been a year of a lack of equilibrium, and most of my talk today will be about balance, because that's what we need in the immune system. And I understand that our students have a very broad background, so I will probably talk a little bit about our own work, but largely in more broad terms throughout this talk. I want to acknowledge that there are many people who have suffered from this disease and continue to suffer from it, and many who have succumbed to it. And we have learned a lot by studying these people, so we owe them a debt. And we have learned a lot, not just about COVID-19, but about human disease in general and about the human immune system. So my next few lines, I call it, "The center cannot hold." Immune cells, they tell their stories through signaling cascades. Their tales are never one-sided. A fine balance pervades. What goes up, it has been said, must someday come down. Moderation is the key. There's no need to go to town. Our species, it was naked. Faced a foe it had never seen. No antibodies in any human to the COVID spike protein. An immune system that is balanced, that generally is the norm. But sometimes immunity goes berserk and the virus starts a storm. Remember William Butler Yeats, but things don't always fall apart. It's when that fine balance is lost that signals go off the chart. Uncontrolled gene expression or a failure of inhibition, and the immune system loses its exquisite precision. That balance, it is crucial, homeostasis, if you please. The loss of immune precision is the basis of disease. Yeats said things fall apart, the center cannot hold. Unregulated COVID immunity has sadly culled the old. So let me share my screen now. Today, what I'm going to be talking about is really about imbalanced immunity in COVID-19. And I'll talk about how immune activation drives the disease. We'll talk about the evasion of the antiviral state. We'll talk about the activation of inflammation by SARS-CoV-2. And we'll talk about innate immune defects that increase susceptibility to COVID-19. And we'll end with a little touching on aberrant innate immunity and how it can stymie adaptive immunity as well. So just to place this in context, you all know that this virus has a spike protein, it's membrane bound, it has an RNA genome, it's a positive strand RNA genome, binds to ACE2. ACE2 binds to what's known as the RBD, or the receptor binding domain, which is in the standing up mode when it contacts ACE2. And we also know that, this is from the work of Jeremy Luban, that when you have this mutation that's very common in the population today, the D614G mutation, the RBD can stand up much more readily, as a result of which you have binding and fusion occurring much more efficiently with the current virus that is spread around the world. We also do know that there's a known protease TMPRSS2 that cleaves the head off, and you heard this from Dr. Glausinger, that the head has to be chopped off. There's a spring-loaded fusion peptide which allows the viral membrane to fuse with the host membrane. This is after it binds to ACE2, you get cleavage of the head. But our cells also have a protease called furin. And this virus has got a mutant spike, in a sense, compared to the old SARS virus. It actually has a few positively charged residues that allows furin cleavage. So when late in infection, we get a little bit more ubiquitous infection of cells, even if they don't have TMPRSS2, if they just have ACE2, this is likely because of furin cleavage. So that's another concern. We all know this, and I alluded to this earlier on, that it's, if you neutralize the virus with antibody, it doesn't get into the cell, doesn't bind to ACE2, but the fact that we didn't have antibodies as a species is the reason why this infection's been so bad. Now, eventually, of course, after infection, the virus will bud out of cells, taking the cell membrane with it, and then infect other cells. If you look at a healthy lung, and the lungs have these alveoli, which are air sacs, you breathe into them, that's where gas exchange occurs. A healthy lung, you can see there's nice air-filled sacs when you look at a section through a lung. But if you look at a COVID lung, first of all, you can see the alveoli are filled with fluid and cells, so you can see how it might be difficult for someone to breathe. And the lung itself is filled with immune cells, so the disease cannot really occur without immune cells getting into the lung. And this is a process we call inflammation. Now, inflammation, if you take a normal pneumonia, you'll notice that you have lots of cells that come into the lung during a pneumonia. This is inflammation of the lung. In a heart attack, in a myocardial infarction, also you have immune cells that will come into the heart. In this case, there's no microbe. In this case, there's a microbe. And the main cells that drive this are cells of innate immunity. One cell is called the neutrophil, and the other cell is a macrophage derived from a blood-borne monocyte. And these are the two main cells that you're going to see go up in COVID-19, and which are going to infiltrate tissues and cause inflammation and damage. Now, we divide the immune system into innate and adaptive immunity. Innate immunity happens immediately. We have lots of cells, we have barriers, and then we make type 1 interferons. And type 1 interferons create what we call the antiviral state, and we talk about that. And then you have pro-inflammatory cytokines, like TNF and IL-6 and so on, which drive inflammation. And on the other hand, we have adaptive immunity, in which B cells make antibodies. This is a slower process. You need a single highly specific cell to expand. It can give you real memory. And in order to get a really good B cell response, we need T cell help. So we have B cells and we have T cells. The T cells do two things. They can help. They can help B cells, for instance, but they can also kill infected cells. So cytotoxic T cells, as you heard from Bruce Walker in the first lecture, are important to kill virally infected cells. And so we have both these arms of immunity, but most of COVID-19 is an innate immune disease. And if there's only one lesson I'm going to give you, it's about disordered innate immunity. And we have to think about adaptive immunity as being something that happens a little later on. However, to make a vaccine, you do need adaptive immunity. You do need your B cells to be helped by T cells to make antibodies that will last for a long time. So if I was to just tell you a little bit about innate immune cells, I'm going to give you this broad, big picture view. We've talked neutrophils and macrophages drive inflammation. These are cells of innate immunity. We call these myeloid cells. These are not lymphoid-like or lymphoid-derived. Okay, and what we basically are going to see, these myeloid cells will increase in disease, neutrophils, macrophages, monocytes go up, whereas all lymphoid and lymphoid-like cells, and I've included plasmacytoid dendritic cells here, and I'll explain this in a minute, and cells called innate lymphoid cells, and of course B cells and T cells all diminish, okay? And this is really part of disease progression, and we'll talk about it. So innate immunity has two arms, inflammation and the antiviral state. And you can break this up in a little more molecular sense, that if I have a sensor that sees something from a microbe, so the sensor sees some microbial structure. This could be, the cell wall of a microbe, for instance. It could be RNA for a different sensor. This sensor is going to send two types of signals. One will induce a transcription factor called NF-kappa B, which will make pro-inflammatory cytokines. And the other pathway will induce the phosphorylation of other transcription factors, which will make type 1 interferons. So we have the generation of inflammation and the generation of type 1 interferons. And most of our sensors will do both. Even though we can talk about this dichotomy, this will happen simultaneously. So we have sensors, then we have a response, we have an innate mechanism of protection. Okay? Now, the location of these sensors is relevant to think about. So this is an RNA virus. The RNA has to divide and replicate, basically make more copies of it. When I say divide, I meant replicate. So you're going to have a lot of double-stranded RNA created in the cell. We have sensors, we call them Rig-like receptors, that can sense double-stranded RNA and say, this is not ours, this is from a virus, we need to respond. We also have sensors that sit in endosomes. So now if a cell made a virus and it got into another cell, so like if SARS-CoV-2 got out of an infected cell and got into a target cell like a plasmacytoid dendritic cell, there it would be uncoated in the endosome or in the lysosome, and that's where it would meet a different kind of sensor, which will also tell the immune system there's something around you which you got to respond to. So this is to tell you that in lysosomes and endosomes, we have TLR7, which will see single-stranded RNA, and this is a crucial sensor to respond to SARS-CoV-2. If SARS-CoV-2 comes out of an infected cell, it gets into cells of the innate immune system, this is the guy that will be activated. There is also some possibility of activating TLR3, which is double-stranded RNA, because the cell can rupture, release some double-stranded RNA as the viral RNA was being replicated, and that double-stranded RNA could also trigger TLR3. And this is all in endosomes. Okay? So the sensors in the cytosol, I'm going to mention them a little bit because then we'll see how devious this virus can be. One sensor in the cytosol is called RIG-I. It's basically a helicase, an enzyme, but can respond as a signaling molecule. And it senses the 5' triphosphate. So in an RNA, so our RNAs we'll see have a cap put on them and a tassel put on them. The cap helps us to translate them, but we put the tassel on just to avoid one of these sensors, and I'll discuss that. So we have these uncapped RNAs being recognized by RIG-I or by another sensor called MDA5, and they're going to activate both the antiviral state as well as inflammation. Okay? So what is the reason to make type 1? If a cell is infected, it makes type 1 interferons. The type 1 interferons then bathe all the neighboring cells, and they get signal to the neighboring cells through a receptor, and they tell the neighboring cells, there's a virus around, you better turn on some genes. And so basically the neighboring cells are now prepared to deal with the virus, sometimes to destroy the viral RNA if the virus gets in, sometimes not to assemble the virus along certain pathways, and another way to even stop translation and stop protein being made so the virus can't survive. And sometimes the host cell will die, but it's better to die than to be infected and infect more cells. So this antiviral state depends very much on making type 1 interferons. Now again, I'm assuming that there are some people who don't think that much about RNA. So I just want to mention that if you think of any host gene, the host gene like ACE2, for instance, which is the receptor, DNA is transcribed to RNA, RNA then gets translated, protein gets translocated and goes to the cell surface. Why does our own messenger RNA not activate these sensors? What makes them, what makes these sensors not recognize them? And the reason really is that the sensors are looking for things that don't have a cap and don't have a tassel. Okay, and we call this cap 0 in the literature, that when you have a 7-methyl guanosine cap for messenger RNA, and then we add a particular methyl group on a sugar, and that you can consider to be the tassel, okay, on this ribose. So this is called cap 0, and this whole structure here is called cap 1. And cap 1 requires the addition of the tassel. If you add the cap, one of the sensors is not going to see the RNA. If you have the cap and the tassel, the other sensor can't see the RNA. And viruses have learned how to do this. And in fact, this tassel probably exists just to differentiate between rRNAs and viral RNAs. But viruses get around it, and certainly SARS-CoV-2 gets around all of this. So basically, RIG-I is seeing these 5' triphosphates, and MDA5 needs to have something that doesn't have the cap 1 structure, doesn't have the tassel. Otherwise, MDA5 can't bind. So what does the virus do? The virus has many enzymes. About 1/3 of the viral genome is designed to evade innate immunity. It spends most of its energy just trying to avoid, turning on the innate immune system. So one of the genes, and there are many genes in the virus, I'm just mentioning a few examples, actually helps you make this cap. There are a couple of other genes as well. This is just a non-structural protein of the virus. Another one adds this methyl group to the ribose, which is the addition of the tassel. So even if a little bit of double-stranded RNA was in the cytosol, it's going to be prevented from being recognized as foreign by being modified in this manner. Okay. Now, Rick Glansinger showed you some related figures. This is a more recent figure that, we make these bags. This virus induces, a couple of viral protein induces our cells to make bags, and the viral RNA hides inside the bag and multiplies in it. So the viral RNA, for the most part, is actually protected from being seen by these sensors which are in the cytosol surrounding it, okay? But then what's more interesting is very recently, there's a really beautiful structure, such an elegant structure, like a piece of sculpture that is created by one of the viral proteins. The viral RNA has to get out of this bag in order to go and make more virus. And so the virus makes this structure. This is from a technique called cryo-electron microscopy. And this structure basically, makes a pore. And the pore, we presume, lets the RNA to come out, and so that you can package the RNA and get out. So this is all actually part of evasion. You put the RNA, replicate it in a bag, and then you have a pore to let the RNA come out, and then you have other fallback mechanisms to prevent the RNA from activating innate immunity. So Dr. Glansinger mentioned this as well. This is another structure that came out recently, showing that the ribosome, the site at which mRNA enters the ribosome, is glopped up by this viral protein NSP1. But it also has the ability to bind the 5' untranslated regions of viral RNAs, so that it's blocking the spore and escorting in viral RNAs so they can be translated. Again, there's some dispute about mechanism and so on, but this is really such an elegant way in which the virus is designed, unfortunately, to actually subvert everything that we are trying to do to get rid of it. Yeah? So just to give you the big picture view, what do we think is going on in a patient with a mild COVID disease? First of all, the viral inoculum is probably a little small. The small inoculum gets into your nose, starts to multiply, and you get some virus made. Some cells get infected. Let's say this is the cell that got infected. The virus is clever enough to say, I'm not going to let you, block me. I'm not going to let you turn on your sensors. I'm going to block your sensors. So this cell is not going to make type 1 interferon. It should, but because the sensors aren't being turned on, it's not going to make type 1 interferon. Some virus is going to spill out of this cell. The cells of innate immunity that are trying to protect us now are cells called plasmacytoid dendritic cells. Some macrophages will try to do the same thing, but the PDCs are really good at making a lot of type 1 interferon. There are a lot of these toll-like receptors in endosomes like TLR3 and TLR7, and these plasmacytoid dendritic cells are going to make a lot of type 1 interferon. These blue balls over here. This is going to bathe the neighboring epithelial cells. So in this mild infection situation, the neighboring epithelial cells are all being triggered through the type 1 interferon receptor by type 1 interferon made by a PDC in order to create the antiviral state. And in this way, the neighboring cells are going to be protected. And so you're going to get an infection, you're going to get some protection, you're going to clear this virus. And most of this is the innate immune system. In fact, it's hard to prove that we need adaptive immunity, though adaptive immunity will come into play in a minute or two. I'll tell you about that. So basically what's happening here is you are favoring what you should favor. You're favoring the antiviral state over inflammation. You're not getting that much damage to the tissue. And then you'll finally get rid of the virus. And this person was either asymptomatic or had a mild infection, recovered soon, maybe never knew that she or he was infected. And this is the scenario we think about happening in this context. What might happen in this context, and it does happen, but is it really protective, or is it just happening because it can? Because this is how the immune system works, is, okay, the small inoculum was probably taken care of largely by innate immunity and by making type 1 interferon. Type 1 interferons also activate cells called natural killer cells, or NK cells. And NK cells are lymphoid-like cells. They are an innate lymphocyte, which can kill infected cells. So this is something that might happen because you had pretty good type 1 interferon production by your PDCs. This is all in mild disease. You would also get what type 1 interferons do. They are not just there to tell the neighboring cells. They go and tell the lymph node, make sure you activate your lymphocytes really well. They prevent our lymphocytes as they're activating from coming out of the lymph node, just so they can get properly activated. They do a number of things that activate adaptive immunity. So you can predict, this is a mild disease. You made type 1 interferons. There was no problem making type 1 interferon by other cells. The infected cell didn't do much. So you're going to get an adequate CD8 T cell response. It's not going to be very much because there's not much virus, right? If you don't have enough virus, you don't have a viral antigen, you're not going to trigger this that well, but you're capable of doing it. And so a dendritic cell, this job is to take the viral peptides and antigens, go to the nearest lymph node, find a specific T cell that recognizes something from the virus, cause it to multiply. Those activated T cells now leave the lymph node, get into the circulation, and home back to the nose or wherever the infection was. And if there is some residual viral infected cell, they might deal with it. Now, as I said, this can happen, but there's not very good evidence for it. And certainly there's no evidence to prove that it's relevant. Until there are good animal models used where you can get rid of specific cell types, this idea won't be testable, but this is what we can visualize happening. So again, let's go and think, we thought about mild COVID. What about severe COVID? I think what you want to think about in severe COVID is that here inflammation is out of control. For what happened is what you wanted to do, you weren't able to do well. You evaded the antiviral state. The virus evaded the antiviral state, but the virus is there and it's multiplying and it's activating cells, and then inflammation went out of control. It just have so much of an inflammatory response. You don't have the antiviral state. You have this imbalance where one arm of the innate immune system, which is particularly good to deal with fungi and bacteria, it can help with viruses, but is a minor component of protection against viruses. So that arm is now going blazing guns right now, whereas the antiviral state, which should be turned on, the virus has very cleverly evaded, and you can't do much about it. So this is the big picture view of what's going to go on in severe COVID. Now, of course, there are some mechanisms that make this happen and make it worse, which is what we're going to consider. So then the first question is, what is it about the virus that triggers these mechanisms? What is it that makes you actually have these damaging responses happen? What can it be in the virus that is triggering us? What is the driver? So we call the molecules in a pathogen that activate innate immunity, we call them PAMPs. So they are pathogen-associated molecular patterns. And all these sensors I'm talking about are just receptors which recognize these patterns from pathogens, and they respond. And that's what innate immunity generally does. So then what are the PAMPs? Obvious PAMP is RNA, right? This is RNA, it's an RNA virus. You look at the virus, the main protein on the outside is the spike. There's a protein called the M protein, and there's the N protein, the nucleocapsid protein, coats the RNA. Okay, and what is happening in our cells is the virus got in, it made a positive strand to a negative strand, used the negative strand as a template, made a lot more positive strand, and packaged more virus, and those viruses got out of the cells. But the main PAMP that this virus is going to produce is still viral RNA. And it's still going to activate inflammation, even if it can't turn on type 1 interferons. The other PAMP that should be considered, and the literature on this is somewhat limited, but I can tell you about it, especially if people have questions, is this nucleocapsid protein of the virus. Okay. This nucleocapsid protein is also capable of activating innate immunity. And the last protein that I think we should consider being as part of what is triggering innate immunity as a PAMP is the spike protein. The spike protein is trimeric, and the spike protein of the virus there's good evidence now, can activate innate immunity. So all of these could be doing something, but on top of that, there's another something that happens in the immune system. If you damage cells, you do surgery, right? Why do you get an immune response after surgery? You didn't have any bacteria. Part of it is because you damage cells. And the damage to cells can release what molecules we call DAMPs. They are things that come out of our own cells that can activate the innate immune system. So because you have viral infection, you have cells that are, going to be quite sick because of viral infection and blocking blood flow and things like that, you get a lot of DAMPs. So these DAMPs are also driving the inflammatory process. So this combination is going to drive inflammation. At the end of the day, you're going to have a lot of neutrophils and macrophages entering your tissues, like your lungs. You're going to make a lot of cytokines, and this is what we end up calling the cytokine storm. Okay, these are pro-inflammatory cytokines and chemokines that are being secreted. So we have two really interesting stories that have come out in the last few weeks, which give us the first good feeling for why some people are susceptible. Okay, we say older people are susceptible. We say if you have type 2 diabetes, you have hypertension, you're susceptible. We don't have a really good feeling other than in age, maybe adaptive immunity may not be that strong. I'm going to say that maybe there's another reason. But these two pieces of data from Jean-Laurent Casanova actually give us a good sense of why some people are getting this disease and getting it severe. So one thing that he showed was about 4% of patients who get severe COVID, and this is a large number of patients studied, about 4% of people with severe COVID-19 and 0 in people who have milder or moderate disease have mutations in entire pathways which are involved in type 1 interferon generation. So having been born with small mutations, these are the polymorphic changes sometimes, but with mutations that affect the function of the type 1 interferon system and cripple it, are now making you more susceptible. Even more interestingly, about 10% of all people with severe COVID-19, they spontaneously make autoantibodies. They make antibodies against type 1 interferons. So in them, even if they made type 1 interferons, even if the PDCs responded, the type 1 interferons can't do anything for them because they've got antibodies blocking it. So this adds up almost to 15%. So almost a sixth of patients with severe disease you can explain by these mechanisms. And so these papers have been very interesting to say, okay, finally we have some handle on understanding why you get severe disease. Okay. So if we think about why do viral PAMPs not trigger the protective antivirus trait through PDCs? I mean, okay, all very well, you didn't have type 1 interferon production or something else. What about the other people? And why don't you get some activation of PDCs like in a mild person? And so one thing to note, and this is something that if we had looked at the literature in mice, we would have known this for a long time. This has been there for a long time in mice, and this is true in human infections as well. When you have a severe infection, when you have, even when you give a lot of an adjuvant into a mouse, you make a lot of inflammatory cytokines, and then this diminishes bone marrow production of lymphoid cells, not of myeloid cells, only of lymphoid and lymphoid-like cells, okay? And thymus production of T cells. And this is understood in terms of the cytokines that drive it. Clearly, much more needs to be done in terms of deeper mechanism, but this is a well-described phenomenon. We can show you papers that go back 30 years showing this time and time again. Severe malaria, this happens as well. So PDCs go down. Now, are PDCs lymphoid-like? We showed this last year. We published a paper showing that PDCs have a different origin, which is lymphoid-like. Florian Guinho's lab, also using completely different method, also showed that PDCs are lymphoid-like. Okay, so PDCs really belong to this group. They're not myeloid cells. There's something we had a notion of before. There's now acceptance that this is not the case. NK cells, which can kill virally infected cells, they also go down. Our B cells and T cells, they also go down. Now, in one of our recent studies, we actually looked to see whether the earliest B cells coming out of the bone marrow, whether they were being lost in this disease. That would tell us there's a bone marrow failure, because we weren't able to do bone marrow aspirates at that time. And sure enough, you get severe COVID, you lose these cells called transitional B cells. These T1 and T2 cells are markedly reduced in COVID-19. Okay, so in COVID-19, just because it's severe infection, a lot of cytokines being made, this is having the effect of also suppressing your ability to produce, an antiviral response by multiple means, including PDCs and NK cells. NK cells actually, I've heard from Jeremy Ruban, they increase with age, but this is now, they're all going away in COVID. You can't find them even in the lungs now. Okay? The other thing that happens is that you make clots. Okay, so there's a hypercoagulable state. And so in different organs, as you get severe disease, what happens is you tend to make small blood clots called a microangiopathy. You will have clots forming in small vessels, can affect everything from your kidneys to your lungs to your heart. And so why does this happen? The virus itself, so just viral infection, when that happens, and this is not always the case, because don't always have viral infection. Like I just heard a talk today about the kidney, and viral infection doesn't seem to happen in the kidney, but you have this phenomenon. But viral infection can damage the endothelium. And then there's a protein released from tissues whenever we damage the endothelium. Endothelium is the lining of blood vessels. When you damage the endothelium, this protein called tissue factor comes out and it sets up a clot. So platelets collect, you form a clot. We activate complement. There's a complement system as part of innate immunity. It's there to protect us, but we aberrantly activate complement. So this virus will activate complement. And by activating complement itself, if you make too much of these factors, can damage our own cells. And that will also drive you towards clots. Part of this process of activating complement and other PAMPs can activate neutrophils to form structures that are called nets, for neutrophil extracellular traps. So these neutrophils, what they do is they get activated, they release their DNA, they form a little mesh around them, and they're trying basically to trap bacteria, put out the contents of the granules which have enzymes that can kill bacteria. But you don't have a bacterial infection here, and you're doing all this, and you're forming this gooey mass inside your blood vessel of nets. And all of this is propagating the formation of clots. On top of it, the complement system, it's a cascade of proteases, also activates the coagulation system. And then your liver is making more coagulation factors because that's part of our innate immune response. As a result, you have this hypercoagulable state. You often now have blood clots forming in tissues, and you get organ damage, not just because of the virus, but because of all these other effects of having, blood flow is reduced, organs don't function, and we have damage. Okay? I'll go through this very quickly because most people can't stand talking about complement, but I'll just briefly mention this to you. How is complement activated? Here, the spike protein is our PAMP. Okay, so normally, all you need to take from this is this message. Normally, our cells have something called heparan sulfate on them, and the heparan sulfate binds to an inhibitor of complement called factor H. Okay. When this binds, we don't deposit complement on our own cells and we are protected. When the virus comes around, SARS-CoV-2, the spike binds really well to heparan sulfate. So it just lodges itself on the heparan sulfate enough to displace some factor H here and there. Now you displace factor H, complement gets activated, you cleave your complement factors, and now it's out of control. Okay, so the virus directly is using the spike protein as a PAMP to activate the alternative pathway of complement. There's a report, it's not yet accepted, so that's why I'm not emphasizing it, showing that the N protein, the nucleocapsid protein, can also activate complement by a slightly different mechanism, okay? And when that is published, there's been online for a few months. I'm waiting to see that that gets, goes through peer review. But that is also possible, that the N protein can activate what's called the lectin pathway of complement, and you get more complement activity. And that's enough to explain most of the phenomena that we're seeing. Okay? So just to summarize this, we have an imbalance. We have more inflammation, diminished antiviral state. We have more myeloid cells, diminished lymphoid-like cells. We have more pro-inflammatory cytokines. We have less type 1 interferon. We have more complement activation, nyctosis, coagulation. We have diminished tissue integrity and organ function. And all of this is coming together to make this a very bad disease. Okay? So finally, for the last 2 slides, I'm going to tell you that COVID-19 and COVID-19 specific innate changes in lymph nodes, which we are very interested in, lymph nodes are where you start your immune response, might be impairing adaptive immunity in a couple of ways. Okay? And this is what I'll just describe in the last couple of slides. So this is something that we reported, and you heard last week from Michelle Nussenzweig about germinal centers. And germinal centers are basically the schoolyards of the immune system, where you make really nice, strong immune responses. You generate memory for B cells. You use this entire system of making a germinal center as a tool to refine your antibodies, make them bind very tightly, and also create memory. Now, we looked at patients with late disease and early disease. And what I want to emphasize is, when we quantitate all this, which I'm not going to show you, you lose your germinal centers. Early in disease, you can see this is what a germinal, this is BCL6 staining over here. BCL6 is just a transcription factor, is really showing you what a germinal center should look like in a normal thoracic lung. So these are controls. Unfortunately, patients who passed away but without COVID-19, and they're age matched and so on. And these are also elderly people, but they have very nice lymph nodes, germinal centers in their lymph nodes. Early in disease, they're gone. Late in disease, they're gone. When you quantitate them, there's really very little, but there's some attempt to come back. So you can imagine that if someone actually survives a severe disease, they might eventually get some germinal centers going, and that might be how you might get a little bit of memory in people who survive a very severe infection. We have explained why this happens by showing that the specific type of T cell that makes these germinal centers, that help you to make these germinal centers, the differentiation of those cells is blocked. Okay? And this is all now published, so I'm not going to emphasize it, but that gives us the best explanation for why this happens. We knew from mouse studies, from other people's mouse studies and other different disease infections, that if you lost your germinal centers in a severe infection and you blocked a cytokine called TNF, the germinal centers came back. So this led us to look inside the lymph nodes. And so if you're now looking at lymph node, this is a normal lymph node. This is the control. And in the control, you can see we're staining for TNF alpha. It's just a little bit of TNF alpha in the lymph nodes. Okay, very little, all in one area. This is what you see normally. And this is what we've known for a while. If you look in the disease, TNF alpha is all over the lymph node. There's a huge amount of TNF alpha in the lymph node. If you quantitate this, the number of TNF alpha cells early in disease and late in disease in lymph nodes is quite a lot of it, but it's really low in controls. So we think this is doing 2 things. One is this is likely the cause, though we still have to prove this by mouse experimentation, of blocking the differentiation of this T cells, the T follicular helper cells that help you make germinal centers. So too much TNF alpha, because you have too much of a type of immune response, we call it type 1 immune response, making these cells make a lot of TNF alpha, blocks the differentiation of T follicular helper cells, and we are looking at the molecular mechanism of this process right now. The other thing that is most likely happening, and we do have some interesting data, both from lungs and lymph node, is that we think CD8 T cells, the numbers are down already because of lymphopenia, but those CD8 T cells in the lymph node, they can't divide, and they're actually being induced to death, to die, by a process we call activation-induced cell death, which in CD8 T cells is driven by TNF. So at the same time, innate immunity from the virus is blocking the differentiation of the type of T cell that helps you make good antibodies, and it's also blocking the type of T cell, allowing the cells to die, the type of T cell that will get rid of virally infected cells. Okay, so it's a fairly pernicious virus that is doing multiple things to actually defeat us. So the big picture view of this story that we've reported is that if you had measles, then you have nice cells that can, the TFH cells that can drive the formation of germinal centers, and then you will make good memory B cell responses. If you get COVID-19, for mechanisms that we've talked about, you have a lot of this TNF production in the lymph node. You don't get the formation of these T follicular helper cells, and you don't get the formation of germinal centers. And as a result, you probably will not, in these patients, generate very good memory. And you certainly don't develop the type of mutation that Dr. Nussenzweig talked about last week, that is part of getting to a high affinity, good antibody response. Okay? So this is just the last slide. This is the big picture view. Severely ill patients, you block, you evade the innate immune system, you have viremia, your immune system, the activation in the lymph nodes can occur, but it's not of the right type. And you have maybe lymphocytes contribute to making the disease worse late in the disease. So you have lymphopenia, you deplete CD8 T cells, your germinal centers collapse. So both arms of adaptive immunity are being crippled by this virus, working through this inflammatory innate response. So I talked only briefly about their work, but there are a number of people at the Reagan Institute who are deeply involved in the work that I've talked about, even briefly. Naoki Kaneko, Xiaoshuan Kuo, Julie Bocco, and Jocelyn Farmer. And our key collaborator, I should mention, was Robert Padera in the pathology department of the Brigham, who provided us with all these interesting samples, and we had wonderful discussions with them. And we had a number of contributors that I can't name all of them at the Reagan Institute who helped us in these studies. And I use BioRender to make my slides. I can end and take questions at this point.
Thank you very much, Chief, for a beautiful talk, and I will be channeling the questions. We got several from the chat. I mean, Miriam is asking, how preserved is the immunosystem across animals? And she wonders whether there are any hints about the host of the virus evolved on the basis of the strategies used to evade immunosystems in the past.
Yeah, so I mean, unfortunately, we don't know the intermediate host for this virus. And there are studies in bats. So it starts from bats, probably goes to an intermediate host. I think that's what the question is about, and then comes to us. And some studies in the bat basically show that the virus can hang around in the bat without causing inflammation, without being, without causing the immune system to get rid of it, but without causing the death of the bat either. So there's been this state of, equilibrium reached there, where the bat is not bothered by the virus, and the virus can multiply, and so it can come into other species.
But that is what Miriam is wondering. She's wondering whether, do bats have this innate response, or do they have the modules to recognize, and can you determine that from the genome of the virus?
So the limited studies that I've read, some papers on the bat, but they were done with an earlier virus, so not with SARS-CoV-2. And in those studies, they basically show that many coronaviruses in the bat, they can evade innate immunity and the bat shuts it off. So they do have innate immunity. They have a lot of those things, but they don't turn on inflammation as potently either. So the exact mechanism, because the bat doesn't have as many reagents for people to study it in the kind of depth that we would like to see, we don't have that kind of granular detail about mechanism. Because it's hard to study species against, other than the mouse, monkey, and humans, we just don't have enough reagents to go in depth. But someday that'll get done.
Then Max is asking, he's curious about how you might address disease and whether you can only from the innate perspective, and how might you approach if there is not adaptive immunosystem. And that is some topic that came in the questions to vaccine development too. In other words, can we deal with this just with adaptive?
So let me be very polytheistic in my thinking, okay? You cannot manage pathogens just by one aspect of immunity. we do need innate immunity and we do need adaptive immunity. And we have beautiful examples, I mean, I would say unfortunate examples in nature, where you have a mutation in one or the other component that we're interested in, you get an immunodeficiency disease. And we know what pathogens are going to come up based on what you lost. So different arms of immunity are more potent in dealing with certain pathogens, but overall you need everything, right? If you lost one, you wouldn't be able to deal with it. So I was trying to emphasize that most of what we see, how do we deal with COVID-19 early on? That's probably innate immunity. What causes most of the problems to us? Okay? What causes most of our problems is that the protective arm of innate immunity, we are unable to use because either the virus cripples it or inflammation cripples it. And the non-protective arm in the context of viruses, this is a very important arm otherwise, right? What is going to be really useful in some situations, if you go out of control, it's just imbalanced. Instead of making X amount of molecules, you're making 10X now. Okay, so you have this out of kilter inflammatory response just because the brakes have been lost, because you kept making more and more virus, and the break here was to use the antiviral state, which failed you. So now inflammation just kicks out strong and strong. It doesn't know what's going on. And that is what's causing most of the disease. Now, this is not to say for protection, absolutely need adaptive immunity. Innate immunity cannot protect us long term, even though we have some kind of memory, which is basically, in innate immunity, that's just epigenetic alterations, which we call a kind of memory in innate immunity. It does not compare with adaptive immunity, where if you get measles, you will be protected for 80 years or 100 years if you live that long. And there's evidence for that from measles, because you made antibodies and you made memory, and those are really long-lived. So we need adaptive immunity, but this disease, what I'm trying to emphasize, is that most of the damage is coming from innate immunity. Now, adaptive immunity may enhance it.
And in a related question, Shiv, I mean, if you are exposed to a limited, a question Richard has put, if you are exposed to a limited viral dose where innate immunity may take care of the virus and there is not much acquired, is this the case in people where, they might live as susceptible because, I mean, innate immunity will deal just with the virus and you will not develop adaptive, and therefore that make you more easy to be reinfected by the virus?
So yes, that is possible. So the point that you're making is that if you actually, so I think the people who are being protected, actually saw data yesterday from Patrick Wilson, who, I heard about the data from Patrick Wilson from a meeting I was at, and Facundo knows him well. So basically it seems that you can make memory in people who are going through severe infection and convalescence. There's no proof of memory, but at least there's proof of some somatic hypermutation. When you have a severe infection, you survived, you went to convalescence, then you start to make a little bit of this potentially memory creating response. That type of response doesn't happen with very mild infection because you just don't have enough virus to create the response. And in severe infection, especially if you don't really get your immune system going and you had, say, a type 1 interferon defect, it can kill you or it can cripple your in adaptive immune system, but maybe you will recover. But by then you might have lost the virus as an antigen, so you may not make such a good immune response. We have virus that's heavy in the first week, and then it declines after a couple of weeks.
And in related to that, do you think that people that get exposed to a low inoculant of virus or a low number of virus may just deal with, I mean, a lot of discussions has been going on about infected with high load or low load. Do you think that in those cases adaptive? These are another question that the students are raising.
Yeah, so I actually do think that if you have a very low viral load, you probably don't get much of an adaptive response just because you don't have enough antigen dose and it gets cleared off. So you just don't have it. To make a good adaptive response, you need the virus to hang around a little bit, or you need an immunogen to hang around a little bit. That's what a vaccine will do for you. So I actually do think that if you're completely healthy and you have a very low level infection, very low viral load, the most likely you will clear it. The studies do show that. As you go to more severe disease, the viral load goes up. So I think that's what happens. I mean, again, this is all surmised from data that we correlate, that you get a mild infection, you didn't have a very high inoculum, you may not get severe disease and you may not get a good adaptive response either. So definitely don't hope for herd immunity for mild infection.
In relation to that, the notion there, so you will agree with the notion that in the absence of disease and memory, is that people will not get this long-lasting immunity.
So I think the long-lasting immunity may come in a small fraction of these people who survived it. And let's say the FDCs or something held on to some antigen and they were able to get back a good germinal center response during convalescence and had enough antigen to go further. But for many people, it won't. And this may be the explanation why that even in SARS and MERS, there were a few patients who had antibodies for like 10, 12 years. But the vast majority, it all vanished by 1 year, even though they had severe disease.
And I have two other questions related to the length of the genome, Shiv. I mean, you mentioned proteins that could be recognized, but the students are asking, why doesn't SARS-CoV-2 cover sepsis, inflammation, and sorry, this is not, can you speak a little more about the third of the genome that does not evade the immune system? What does is for?
Obviously, so you need to make the virus, you need to replicate the virus, you need to have the spike protein. It's really to enter cells to get infected. So the virus, basically, if you divide the genes of the virus, there are those genes which are structural, those genes that allow the virus to divide, which is nothing to do with evading to actually form more virus, which has nothing to do with evading innate immunity. And then there's all those genes which help it to hide inside the cell or not be seen by a sensor and not to respond. And those are all for immune evasion. And without those, this virus would not be a pathogen. It would be just a harmless virus that you just lose every time.
And then why does SARS-CoV-2 cause excessive inflammation? It doesn't cause excessive inflammation in all the patients. You alluded into some mutations during the talk, but and this is because only related to the viral load, or is?
So, I mean, obviously we don't know all the answers, but I'll tell you what we do understand. So we understand one thing, yeah, low viral load, you're probably fine. But if you had a low viral load and you had a mutation in a type 1 interferon gene, then I don't know, you'll end up with a high viral load, right? You just want to respond, you want to protect yourself, you might end up with a high viral load. So it's all depends on how we look at that. So it can't be just low viral load in everybody. In most people, low viral load, you'll be fine. If you're susceptible because you already made antibodies to type 1 interferon, or you have a mutation, or for the reason that we don't understand, why is somebody who's obese more likely to be susceptible? I don't have a good feeling yet. I mean, they can postulate things that might be the case. Then maybe a low viral load will become a high viral load because you didn't get protected perhaps, and then it expanded more. But in general, a low viral load will end up with mild disease, and a higher inoculum will end up with higher disease. That's why if you look at doctors who in the early days when they didn't have proper protective gear, we had a lot of very young doctors in certain parts of the world, including the US, get infected, and also not in Boston, but in other cities. We had intensive care nurses who got infected and got the disease. And the reason they probably got the disease is they were so exposed, they got high viral loads.
And then there is a whole set of other questions that we don't have the time to address. I just would like to thank you very much for a beautiful talk, and also put you a request from the students. They would like to know where your poem is published anywhere, and otherwise they would like to get the words sent to them if you can. So
okay,
very much.
Just to the students, and one day I'll publish it, maybe, who knows, when I get time.
Thank you. Thank you, Shiv, for a wonderful talk, and thank you all for attending this and the interesting questions you have raised. Thank you very much.
Thank you very much. Thanks for the invitation.